Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Now, this is my third video lecture in my series on photosynthesis. Now, the first one I talked about, why we care about photosynthesis, and the second one was about how do they actually capture light energy? And today, what we're going to talk about is the light reaction. So let's start with the big picture first. Photosynthesis, putting together by light. Photosynthesis uses the energy in sunlight to fix carbon dioxide into organic molecules. So we're going to take carbon dioxide, we're going to take some water, and we're going to use the energy to basically rip those electrons and hydrogens from the oxygen and water, okay? And we're going to put them into glucose, and this, of course, is going to release some oxygen. So the overall chemical reaction for photosynthesis is summarized like this. We take 6 carbon dioxides. That means we have 6 carbon atoms, right? We also take 6 water molecules, and we use that input of energy from the sunlight, and we're going to fix carbon into organic forms by adding hydrogen to it and by adding electrons. And then, of course, we're going to release oxygen as a byproduct. Now, this is important because this is a redox reaction, reduction oxidation. In this case, carbon is going to go from an oxidized form to a reduced form. It's gaining electrons. It's like gaining energy. And oxygen, of course, is going to be oxidized from its reduced form in water to an oxidized form as molecular oxygen. So I've already said this, but you need a source of electrons, you need a source of energy. And of course, the source of electrons is from water. So we're going to talk a lot about the flow of electrons and how they're used in the light reaction, and will ultimately end up in the Calvin cycle. We'll talk about that in a second. But because we're making a larger, more complex molecule, like a glucose molecule. Now, just to be clear, the Calvin cycle doesn't actually make glucose, but it does make organic molecules. And by fixing carbon dioxide into organic molecules, that's an endergonic reaction. And you need energy for an endergonic reaction. So the Calvin cycle, which we'll talk about here in a second, so for this endergonic reaction, we're going to need a source of energy. And of course, that energy is in sunlight, and parts of photosynthesis transform the kinetic energy of sunlight, specifically electromagnetic radiation, into a chemical energy that can be used to now fix carbon dioxide into organic molecules. And of course, endergonic reactions, you can see that the products have more potential energy than the reactants. Okay, so what I want to do now is just give you an overview of photosynthesis. We know we're going to take carbon dioxide, right? And we're going to fix that into an organic molecule. But let's talk about the big picture, and then I'm going to go into the nuts and bolts of how the light reaction works, and also photophosphorylation as well. So let's review the whole process really quickly. You've got 2 parts. You've got the light reaction. This is the photo part. This is where you take incoming energy from sunlight and transform that into chemical energy. And we're going to do it in 2 different ways. We're going to reduce an electron carrier, NADPH, to, well, that's the reduced form of it. And we're going to make ATP. Both of those have chemical energy. And then the second part is the Calvin cycle. And the Calvin cycle is often called the dark reaction. I don't like that. It's the synthesis part. The Calvin cycle is where you actually take carbon dioxide and fix it into an organic molecule using the electrons and hydrogens that you got from water and the energy you got from sunlight. And of course, the light reaction. Okay, light reaction is very important. It's a transformative reaction, right? It's taking energy in sunlight. That's what the chlorophyll molecules do. And it takes the energy and the electrons and elevates them in energy. Because when you're pulling electrons off of water, they don't have a lot of energy. So the light reaction energizes those electrons, and they're used to make ATP, and then they're used to basically reduce NADP to NADPH, okay? And it's also going to give us a source of hydrogens, because we're going to rip those hydrogens from water, and of course those hydrogens get bolted onto carbon atoms so they can become an organic form. But we also use those hydrogens in chemiosmosis and photophosphorylation. So that's really, really cool that these electrons and the protons both have a couple different purposes here. Now, of course, we're talking about the photosynthesis in plants. And of course, we know that photosynthesis inside of plants takes place and the chloroplast, which were once upon a time ancient cyanobacteria. Wild. Well, photosynthesis is a fairly complicated process, right? It requires a series of metabolic reactions, and it requires specialized structures. And those specialized structures are the thylakoids. And thylakoids are these flattened sacs, okay? And they have an interior. It's called the lumen of the thylakoid. Anytime you see the word lumen, it just means the inside of something. It's also called the thylakoid space. And you've got a thylakoid membrane. And the thylakoid membrane, when you see a chloroplast under a microscope and it looks green, what you're really seeing are thousands and thousands and thousands of these thylakoid membranes that have the chlorophyll embedded in them. So the chlorophyll is embedded in these thylakoid membranes, and this is where the light reaction takes place. And the light reaction, of course, it splits water, releases molecular oxygen, gives us a source of electrons to reduce carbon dioxide later, and it also transforms electromagnetic radiation into chemical energy. Now, let me say this a little bit more, less technically here. It's converting kinetic energy to potential energy. So remember, electromagnetic radiation is a form of kinetic energy because these photons are moving in waves. And then, of course, the second part is the Calvin cycle. This is the synthesis part, and this is where we're going to actually fix carbon dioxide into organic molecules. And the Calvin cycle occurs inside the stroma of the chloroplast organelle. Now the stroma, okay, don't confuse that with the stomata. Stomata are tiny openings on the underside of leaves that allow for gas exchange. That's how water exits leaves, oxygen exits leaves, and carbon dioxide comes in, okay? But the stroma, this is the area between the thylakoid membrane and the inner membrane of the chloroplast. And this is where the Calvin cycle occurs. So once you don't have sunlight, most of the time the Calvin cycle will stop. Okay. And of course, it pumps out organic molecules that can go on to be made into sugars and any other what is called secondary metabolite that the plants make. Now, secondary metabolite. Secondary just means second, right? I mean, it's pretty straightforward. Metabolite. Metabolism. Metabolism is making things, right? Breaking is chemical reactions. So a secondary metabolite is any molecule the plant makes basically after making sugar. So any type of nucleic acids, lipids, hormones, those would all be secondary metabolites made by plants. Okay. And importantly, the flow of electrons, ultimately starting with water, right, going all the way to the organic molecules, those flow of electrons link the Calvin cycle to the light reaction. So as you realize, the Calvin cycle cannot occur without a supply of energy and the supply of electrons coming from the light reaction. Let's narrow it down a little bit. We're going to not talk about the Calvin cycle here. We're going to talk about the light reaction. The light reaction takes place on the thylakoids, okay? Now, light reaction, I've already said this. We're going to take electromagnetic radiation. You're familiar with it. That's just light. That's a light we see, right? And we're going to convert that form of kinetic energy into potential energy. And specifically, that potential energy is going to be chemical energy stored in both ATP. Okay, we're going to make ATP from the light reaction, and we're going to have those high energy electrons. So what the light reaction has to do is energize the electrons, right? Well, actually, it's got to strip the electrons from the water, which needs energy to do that, and it's going to elevate them. So we're going to elevate these electrons, and those electrons, when they first get elevated, they're going to go down an electron transport chain that has a proton pump to create an electrochemical gradient that powers ATP synthase through chemiosmosis to make ATP. I think we've seen that before. Cellular respiration, oxidative phosphorylation. Yeah, isn't that amazing? Plants make ATP really similarly to the way cellular respiration makes ATP. We're going to come back to that. So I'm going to plant that seed. Keep that in mind, because to me, this is just nature repurposing, something that works really well into something different. So as we start taking a closer look at the light reaction, remember this of course is occurring on the thylakoid membranes, we have these pigments, chlorophyll. And what the chlorophyll does is it absorbs sunlight, and then it takes that energy and it elevates electrons into a higher energy state. So basically, you've got this energy coming into these chlorophyll molecules. A whole bunch of these chlorophyll molecules, like 2 to 300, they form an antenna complex with a reaction center at the middle of it. And it transfers all this energy from all these different chlorophyll molecules to the reaction center, where it can then send an electron way high up in energy, where it's picked up by an electron acceptor, specifically phaophytin, which we'll be introduced to in a minute. So at that point, once the electron acceptor picks up an electron, that electron has energy, we've transformed the kinetic energy of sunlight, electromagnetic radiation, into potential energy stored in these high energy electrons. Now, of course, in my previous video, about how pigments absorb sunlight, this random walk that they show, that's, this inductive resonance is what we typically think of as how they transfer the energy without the electrons actually moving place. We know that there might be some quantum coherence going on there, some little quantum weirdness, where these electrons are going everywhere all at once. They find the reaction center, then they become decoherent, and then the electron is in the reaction center, and they can do this very quickly. And that would explain the 98% efficiency of chlorophyll molecules. It's really remarkable. Okay, now, so we got chlorophyll molecules that make up an antenna complex, okay? Now, an antenna complex is part of a larger molecular complex called the photosystems. Now, there are two photosystems that we'll talk about. This is in plants, right? There's Photosystem II and Photosystem I. Now, wait a second. Why did I say Photosystem II first? Well, the reason why is Photosystem II was discovered after Photosystem I. Even though when we start talking about photosynthesis, we start with Photosystem II. I know, it's confusing. It's just in the order of which they were discovered. So here's what these photosystems do. They are these large molecular complexes, and basically they're taking light energy and they're using it to elevate the energy in the electrons. Now you're probably thinking, electrons? Well, where are the electrons coming from? And you probably remember, I've talked about this before, they come from water. Okay, so let's start with Photosystem II. Look at that diagram. Photosystem II is a large, complex molecule. It's got 20 different subunits, 99 cofactors. It has various pigments, electron acceptors, lipids, the antenna complexes. I mean, all of these things go together to form this really large photosystem II. It's a large molecular complex. And like these photosystems, what it does is it elevates electrons and energy. But photosystem II does a couple things differently. One thing that is important for is splitting of water. Now you're probably thinking, whoa, we're splitting water? We're ripping electrons away from oxygen? That's hard, right? It's the second most electronegative element in the universe. So if you're going to rip an electron away from oxygen, you need some energy. And there's also some complex biochemistry that occurs, but we're not going to get into that in this lecture. But what the photosystem II does is it's using the energy in sunlight along with some enzymatic action to basically split water. We're ripping the electrons away from the water and also the protons too, the hydrogen ions, right? Because we need those. Because in the overall reaction of photosynthesis, okay, we're going to take carbon dioxide and reduce it into organic forms, which means we need our electrons, which are coming from the water, and to make organic carbon, it has to be attached to at least one hydrogen. And hydrogen is also coming from water. Now, you're pulling an electron away from oxygen, it has almost no potential energy, right? It has no energy to do work. So it elevates the electron and energy. That is important, right? So that's what the photosystem II is doing. It's splitting water and then it's elevating the electrons in energy. It's elevating them in energy. And it's also releasing molecular oxygen that just basically diffuses out of the cell. Okay? Now, once the photosystem II elevates the electron, that electron is picked up by an electron carrier. And we're starting to see this here. We're going to go down an electron transport chain, right? And look at this carrier. It's called phaophytin. Looks familiar to me. It's got an isoprenoid tail. It's got some ring structures. Some nitrogen in there. Wait, it's missing something. It's missing magnesium. This is basically a chlorophyll molecule. So our first electron acceptor that picks the electron up from photosystem II, this is basically a chlorophyll molecule missing a magnesium ion. Interesting, isn't it? But that makes sense, right? I mean, this molecule is hydrophobic. It can easily go through the membrane, the thylakoid membrane. Okay, then this molecule, phaophytin, will take the electrons to another electron carrier called plastocyanin. Quinone, quinone, quinone. It would be quinone, right? You've seen this before. In cellular respiration, one of the electron shuttles is called ubiquinone. So quinones are a class of molecules, and of course plastocyanin is very similar to ubiquinone that we see in cellular respiration. Now we have this electron transport chain. So where's it going to take it to next? How about a cytochrome complex? Wait, electrons flowing down electron transport chain into cytochrome complexes? Oh my gosh, I mean, how exciting is this, right? I mean, what we're having here, the cytochrome complex, this is cytochrome B6F. It's a proton pump. I know, wild. What this molecule, or this large molecular complex does, is it takes the energy in electrons and it pumps protons into the thylakoid space. You can say this with me. It's creating an electrochemical gradient. This is storing potential energy. So as the photosystem II is ripping the electrons and protons off, those protons are then getting pumped into the thylakoid space. We're storing potential energy. That's what the cytochrome complex does. So let's think about our flow of electrons. They started with water. We elevated them high in energy in the reaction center of our photosystem II. They are then picked up by phaophytin. That drops them off the plastocyanin, and now they're at the cytochrome complex, specifically cytochrome B6F, where it's doing work. They're pumping protons. And as you can imagine, since I'm using these electrons to do work, they're losing energy, okay? So before I go on to what happens to these electrons next, I kind of want you to look at this protein complex, cytochrome complex specifically. You know, and you look at it and it's kind of, oh, blah, 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 you know. But think about how complicated that is, right? Each one of those different colors represents a different subunit of this large protein complex. And if you go way back to when we first learned about proteins, do you see all the alpha helices? Yeah, those are almost certainly hydrophobic regions to allow it to embed into the thylakoid space. The other thing that I want you to think about, somebody figured that out. Like, it took a long time to figure out the structure of these cytochrome complexes. And to me, you know, I just find that so interesting that we can figure out the structure of these large molecular complexes and how they work. It's amazing. Okay, so cytochrome complex, you have to shuttle the electrons away once again. Now we have another electron carrier. This is called plastocyanin. This is a very strong oxidizer. It can easily pick up the spent electrons from cytochrome complex, and it's going to do one thing. It's going to take them to photosystem I. Okay, now, photosystem I, it receives these electrons from plastocyanin. They have no energy. Okay, well, we can't use low energy electrons if we're going to fix carbon dioxide. And think about this. We're going to fix carbon dioxide into glucose ultimately. And we know from cellular respiration that glucose has lots of potential energy, right? Those are the high energy electrons that power oxidative phosphorylation. So you just can't walk around with low energy electrons and expect to fix carbon dioxide. So what photosystem I does is it elevates the electrons back in energy, okay? And then from there, what happens is it gets picked up by an electron carrier called ferredoxin. So then ferredoxin shuttles the electrons to another large protein complex called NADP reductase. The name says it all, right? NAD, nicotine adenine dinucleotide. NAD, right? I mean, with a phosphate group. We've seen that before as well. This is an electron carrier. So NADP reductase, reduction, ACE, ASE, this tells you it's an enzyme that's carrying out a chemical reaction. What NADP reductase does is take the electrons from the from ferredoxin, and it reduces NADP to NADPH, and you notice it picks up a hydrogen. Because if we're going to take carbon dioxide and make an organic molecule out of it, not only do we need a source of high energy electrons, we need a source of protons as well. So there you go. That's what it does. Now these electrons are in NADPH, and they will be shuttled to the Calvin cycle, where they will reduce carbon dioxide into an organic molecule. let's take an overall look at this Z scheme. Yeah, I totally see a Z there, don't you? N? Come on, guys. Not you in the class and the people watching it, but the people that came up with the Z scheme. This is an N scheme, right? Up, down, up, down. I love it. This is the flow of electrons in both energy and in space. So in Photosystem II, they are elevated in energy. They are dropped off through the electron transport chain where they do work to create an electrochemical gradient. They lose their energy, they are re-elevated in energy, and they are carried to ultimately NADPH, which is then used to reduce carbon dioxide. Okay, now, one last thing here. Think about this. The Calvin cycle. We haven't learned about it, but we're going to do a series of chemical reactions that are endergonic. And you know from previous lectures that an endergonic reaction, the way we usually do them, is through energy coupling. Right? Energy coupling. Let's think about that. We have an endergonic reaction. It needs energy. So we couple an exergonic reaction to an endergonic reaction. The way we often do that, you can see where we're going. The way we do that is by phosphorylating an intermediate. So that means that we need a source of ATP to create phosphorylated intermediates. So here's how we make ATP. Photophosphorylation. Wow, the name says it all. Photo means we're getting energy from light. Phosphorylation means we're going to phosphorylate ADP to ATP. So the electrons, what they did is they created an electrochemical gradient. They are storing potential energy by pumping protons into the thylakoid space. This is just like oxidative phosphorylation, right? And then the protons, they can't flow through cellular membranes very well, right? So what they do is they push their way through ATP synthase in a process called proton motive force. Protons moving with force. It's also called chemiosmosis because chemiosmosis means to push. And now we're taking the kinetic energy of protons pushing their way through the ATP synthase, spins up the ATP synthase, and it takes phosphate groups and slams them onto ADP, and we phosphorylate ADP to ATP. We call that chemiosmosis. And that's the same ATP synthase used in cellular respiration. Wow. And then the ATP is then used in the Calvin cycle as to create our phosphorylated intermediates. So that's pretty cool, isn't it? Once again, here's chemiosmosis. Now, this should look very familiar. One thing that I always love to ask is what are the similarities and differences between oxidative phosphorylation and photophosphorylation? Well, first of all, phosphorylation. They're both phosphorylating ADP to ATP, and they're harnessing a proton motive force to do that. And to store the energy, they're both relying on electrons flowing through an electron transport chain to pump protons across the membrane and storing that potential energy. That's where they have in common. I mean, even the ATP synthase is almost identical. So they're both relying on chemiosmosis and proton motive forces. The differences: source of electrons. The source of electrons in oxidative phosphorylation comes from the electron carriers. And they ultimately got their electrons from the breakdown of organic molecules and these high energy electrons. Okay? That's also their source of energy. In photophosphorylation, the electrons are coming from water. They have no potential energy, right? Or very little potential energy, not enough to drive the system. So the electrons are coming from water, and ultimately the photosystems are using energy in sunlight to elevate those electrons. So the source of energy here is different. And what that also tells you is that if you go back to cellular respiration, guess what? The ultimate source of energy, the sun. Okay, well, I hope you enjoyed this lecture. Until next time, I'll do the Calvin cycle on my next series here.